Welcome to Chapter 15, the Chromosomal Basis of Inheritance. Firstly, we will talk about Mendel's hereditary factors, which were actually purely abstract when first proposed. Now we know those factors as genes, which are located on chromosomes. The location of a particular gene can be seen by tagging isolated chromosomes with a fluorescent dye that highlights the genes. Cytologists worked out the process of mitosis in 1875 and meiosis in the 1890s using improved techniques of microscopy. Cytologists are biologists who study the cell and its structures. And biologists began to see parallels between the behavior of Mendel's proposed hereditary factors and chromosomes. Around 1902, Sutton and Bovary and others independently noted these parallels and began to develop the chromosome theory of inheritance. Here is the chromosomal basis of Mendel's laws. Earlier, we talked about the different alleles, which are alternate forms of genes. And these alleles are actually going to be located on the chromosomes. So we have the yellow round seeds here and the green wrinkle seeds here. The yellow round are all dominant, while the green wrinkled are all recessive. We can see how the two parents here are going to go through meiosis and produce the gametes, which will fertilize and produce the F1 generation. And the F1 generation will produce gametes through meiosis as well, and eventually produce the F2 generation. The chromosomal basis of Mendel's laws shows that genes are located on chromosomes, and alleles are going to be located on each individual chromosome in a homologous pair. Okay. So here we see the parent generation, the P generation, and we can see how each allele, the dominant alleles, are going to be on the chromosomes, as well as the recessive alleles in the other parent. After meiosis, we go through a reduction of chromosome number, and the gametes are going to have half as much as the chromosomes, which is n equals 23, being haploid cells. The first solid evidence associating a specific gene with a specific chromosome came in the early 1900s from the work of Thomas Hunt Morgan. His early experiments provided convincing evidence that the chromosomes are the location of Mendel's heritable factors. So Mendel really didn't know about genes or DNA back then. He just called them heritable factors, using the purely probability laws in mathematics to come up with his system of Mendelian genetics. So back to Morgan. For his work, Morgan chose to study Drosophila melanogaster, a common species of fruit fly. Several characteristics make fruit flies a convenient organism for genetic studies. So Morgan probably picked fruit flies because they produce a lot of offspring and they reproduce very quickly. A generation can be bred every 2 weeks, and they don't have 46 chromosomes like we do. They have only 4 pairs of chromosomes, which makes it very easy to observe. So Drosophila melanogaster, the fruit fly, was the model system or the model organism used by Morgan. Morgan called the most common and normal phenotypes the wild type phenotype. Traits alternative to the wild type are called mutant phenotypes, the less common. The first mutant Morgan discovered was a fly with white eyes instead of the wild type red eyes. So here we see the wild type, which is the normal phenotype, red eyes, and the mutant white-eyed fruit fly. In one experiment, Morgan mated male flies with white eyes, mutant, with female flies with red eyes, the wild type. The F1 generation all had red eyes. The F2 generation showed a 3-to-1 red to white eye ratio, but only the males had white eyes. Morgan reasoned that the white-eyed mutant allele must be located on the X chromosome. Morgan's findings supported the chromosome theory of inheritance. So in this slide, we see the experiment set up, which is a cross between the wild-type female and the mutant male. And we can see how the alleles are going to be located on the physical chromosomes. So we remember that females have X and X chromosomes, and we can see how the wild-type female has the W plus allele on both of their X chromosomes, while the male has an X and a Y chromosome, and the W allele, which is the mutant allele, is on the X chromosome only, because this is a X-linked trait. Okay. So next we see the parent generations are crossed. The sperm and the egg are going to fertilize, and a Punnett square is drawn, but we only have 2 squares. So we can have 4 squares, so we'll start with that. So if you look at the female here, the genotype is going to start by drawing out 2 X's, and then you're going to put the alleles. So for the female, it was W plus and W plus, and you put the alleles as exponents. And for the male, it's going to be X, W, and Y blank, because the alleles are on only the X chromosomes. Okay, so now that we have the genotypes, we can draw a Punnett square here. And I'm going to put the alleles. So I'll put mom on the top and dad on the side here. So it doesn't matter which order. And we're going to cross them. So W plus is going to come first because W plus is the dominant allele. And it's the wild type. Okay. And on the bottom, we're going to have the males, XW plus Y, XW plus Y. So we know for sure that the males are going to be red-eyed because they have the W plus, the wild type allele. Okay. And then we see the females. They have both the W plus and the W allele. And since W plus is dominant over W, the females are also going to be red-eyed. And that's what we observe in the F1 generation when we cross the parents. They all have red eyes. So we have 2 females that are carriers of the mutant allele, but the dominant wild type allele is going to be expressed over it. Okay. So after that, we can form a cross between the female and the males, the children from the F1 generation, with each other. So if we do that, let's erase that. I'm going to cross again. We had 2 types, right? We had X W plus and X W, and we had XWY males. Okay, so we're going to do a cross between those. So we're going to have XW plus XW. Oh, I forgot to draw the Punnett square. Okay. And we can have XW and Y. Oh, I'm sorry, W plus. W plus. And so if I cross the F1 generation children together, I'm going to get XW plus, XW plus, XW plus, XW. So 1 carrier right there, XW plus Y and XWY. So this time we have a mutant. We have a mutant. And it's going to be one of the sons. Okay. So the F2 generation ratio is going to be 2 females with red eyes and 1.5 males with white and red eyes. Okay. Okay, so these are our possible genotypes: XW plus, XW plus, XW plus, XW, which is a carrier, XW plus Y, and XWY. Okay. So the reason why they did not add 4 squares here is because they did not include the second X chromosome from the eggs. Because both of the X chromosomes have the same W plus. Okay. So Morgan's discovery of a trait that correlated with the sex of the flies was key to the development of the chromosome theory of inheritance. Because based on this experiment, scientists, they concluded that genes are located on the chromosome. And the alternate versions of the genes are the alleles, and the alleles are going to express a trait or characteristic. Okay. So humans and other mammals have 2 types of sex chromosomes. The X chromosome is going to be larger than the smaller Y chromosome. As you can see on the diagram, it's a lot bigger, which means it has more genes. A person with 2 X chromosomes develops as a female, and if you have an X and a Y chromosome, you are going to be considered a male. Okay. So here are some different chromosomal systems based on different species. We're going to mainly focus on humans. Again, this is just an overview of meiosis. We have 44 autosomes and 2 sex chromosomes for a total of 46 chromosomes. And each of the parents, so humans, we have 46 chromosomes. And after meiosis, we're going to split it into eventually 4 gametes. So the sperm can either have a X chromosome or a Y chromosome, and the eggs only can have X chromosomes, along with 22 other autosomes for a total of 23. And eventually, when the sperm and the egg, they unite, it's 23 plus 23. So we're going to get either a male or a female zygote, depending on, really it depends on the sperm. So if the sperm had an X chromosome, then we're going to get a female. If the sperm had a Y chromosome, we're going to get a male zygote. And the zygote is going to eventually develop into an adult through mitosis. Okay. And we see different systems here. For example, for the chicken and the grasshopper, they have different numbers of chromosomes, and also they're represented by different letters. So we'll mainly focus on human. Okay, next we will talk about the SRY gene, which is located on the Y chromosome. The SRY gene is important because it is going to express the male characteristics of the male and also help with the development of the testes in the embryo. A gene that is located on either sex chromosome is called a sex-linked gene. A gene that's located on the Y chromosome is called a Y-linked gene, and a gene that's located on the X chromosome is called a X-linked gene. Only 78 genes, coding for about 25 proteins, have been identified on the human Y chromosome, so that's a very small amount compared to the X chromosome, which contains about 1,100 genes. And that makes sense because the X chromosome is a lot bigger as well versus the Y chromosome. The X chromosome, it's a sex chromosome, but it also has many genes that express many different characters unrelated to sex. Many Y-linked genes are related to sex determination. X-linked genes follow a specific pattern of inheritance. For a recessive X-linked trait to be expressed, a female needs 2 copies of the allele, so they would have to be homozygous recessive, but a male only needs 1 copy of the allele, and that would be considered hemizygous. This is because the male only has 1 X chromosome, the other being a Y chromosome, and so that is the reason why X-linked recessive disorders are much more common in males than in females, because males only need 1 recessive allele in order to be affected. So we have here a cross between a male and a female, and they produce the offspring. So for A, we can see how we have a affected male right here who's hemizygous for the recessive allele. We remember that the recessive allele is the, let's say it's the disorder, whereas the dominant allele is a normal individual. Okay, so in example A, we have an affected male and a homozygous dominant female, but in B, example B, we have a carrier female who has a dominant and also a recessive allele and a normal male. And in the third example, C, we have a affected male and a carrier female with 1 dominant and 1 recessive allele. And we can see that they all produce different ratios of the different genotypes. So even though the male is affected, so in the first example right here, if the female is homozygous dominant, none of the offspring are going to be affected, or at least show the characteristic. Here are some examples of X-linked recessive disorders. We have colorblindness, which is mostly X-linked, Duchenne's muscular dystrophy, which is a mutation in the dystrophin gene, and dystrophin is an important protein for muscles. So patients with Duchenne's muscular dystrophy, they have a severe muscle weakness and hemophilia, which is a bleeding disorder caused by a mutation in genes that express important clotting factors in order to properly clot the blood. Next, we will discuss X inactivation in female mammals. In early embryonic development, one of the 2 X chromosomes in each cell is randomly inactivated. The inactive X chromosome is called a Barr body, and if a female is heterozygous for a particular gene located on the X chromosome, she will be a mosaic for that character, meaning that she will express both of those phenotypes. Here's an example of X inactivation in cats. So this is a tortoiseshell cat that expresses both the black fur and orange fur. And this is due to X inactivation early on in the development of the embryo. So before, the X chromosomes are all active. One of the X chromosomes has the allele for orange fur, and the other one has the allele for black fur. But as you can see, during the development of the embryo, after a couple cell divisions, one of the X chromosomes is going to be inactivated in certain cells, and vice versa for the other cells. So we have 2 populations of cells, and the cells we're talking about in this case are called melanocytes, which are the skin cells that are going to produce the pigment melanin, which is going to determine your fur color in this case. So one cell population has an inactive orange fur
containing X chromosome, and the other cell population has an inactive X chromosome that had the black fur allele. Um, and this is going to cause the cat to have both black and orange fur because of those different populations of cells. Each chromosome has hundreds or thousands of genes, except for the small Y chromosome. And genes that are located on the same chromosome tend to be inherited together and are called linked genes. So if we had a chromosome right here, and let's say we had gene A and gene B, gene A and gene B would be linked because they're on the same chromosome. However, if we had another chromosome, for example, two different types of chromosomes, so right here, okay, I'm going to draw the homologous chromosomes. And yeah, let's draw the homologous chromosomes here. Okay, so on the other hand, okay, so this is possibility 1, possibility 2. Okay, if we had the gene on separate chromosomes, it would look like this. So gene A is right here, and gene B is right here. Well, in this case, the genes would be unlinked because they're on different chromosomes. Thomas Morgan was a famous scientist who did experiments with fruit flies to see how linkage affects inheritance of two characters. So Morgan crossed fruit flies that differed in traits of body color and wing size. The first cross was a parent generation cross to generate the F1 dihybrid flies. The second was a test cross. Okay, so here we see Morgan's experiment with the parent generation of the fruit flies. So the couple of keywords here, wild type is the normal fruit fly, and the mutant is the non-normal one. So wild type would basically kind of be, in a sense, dominant. The wild type alleles are going to be expressed with a plus sign, while the alleles for the mutant are going to not have a plus sign. So B plus is going to be representing a gray body. B is a black body. VG plus is going to be a normal wings. And VG are going to be vestigial wings. So vestigial wings are a lot smaller and non-functional. And so the parent generation are crossed together, and the F1 dihybrid generation are all going to be wild type. However, they're heterozygous for wild type. They have 1B plus and 1B, and 1VG plus and 1VG. And next, with the F1 dihybrids, we do a test cross. So that's part 2 of the experiment. So this is the test cross. And the test cross is usually used to find the genotype of the mystery individual. And in order to do that, we cross them with a homozygous recessive individual, which is our black-bodied, vestigial-winged male fruit fly here. So after the test cross offspring are shown below. And we got the probabilities of the different fruit flies using a Punnett square. The predictive ratios were, there were 2 predicted ratios. So if the genes were on different chromosomes, then we would see 4 different types of combinations of phenotypes. So if the genes kind of moved around, But if the genes were still on the same chromosomes, then we would have only seen two different phenotypes, which were basically the phenotypes that looked exactly like the parents. Okay, but however, we see here we have two new phenotypes. So we have gray and vestigial, or black and normal wings. So those don't look like the parents. Okay. So the predicted ratios were different from the actual results. The actual results showed us more of the parental genotypes or phenotypes, so 965, 944, and less of the non-parental phenotypes, but they were still there and greater than 0. So we're going to go over why some of the offspring, they don't look exactly like their parents. And if you guessed it, based on previous chapters, it is meiosis and what happens during meiosis, which is crossing over. Okay. Morgan found that body color and wing size are usually inherited together in specific combinations, the parental phenotypes. He noted that these genes do not assort independently and reasoned that they were on the same chromosome. Okay, so Morgan thought that the wild type the wild type phenotypes were on the same chromosome for the parents. And that they were going to be inherited in this way. Okay. So here we see that the dihybrid females and the homozygous recessive male, so the dihybrid females had the wild type from their wild type parent, right here, one chromosome from mom and one chromosome from dad. So this was from mom, from the P generation, and from dad in the P generation. Okay. And we have the mutant male, which has both the BVGs. So basically, Morgan thought that these stick together. The wild type and the mutants stay together. And throughout each generation, they're going to inherit either or. Okay. So most of the offspring were going to show the parental phenotypes. And that's because these offspring are going to look like their parents. This one's going to be black-bodied and vestigial wings, which looks exactly the same as the parent here. And the other offspring is going to have a gray body, which was B plus B, and normal wings, which was VG plus VG, and that looks exactly like parent here. Okay, so these are the parental phenotypes, but did all of the offspring look like this? And the answer is no. Some of the offspring had different combinations, and that was possible because of what we know happens in meiosis, genetic recombination. during crossing over, which occurs in prophase 1. Non-parental phenotypes were also produced in the test cross because of this phenomenon. The genetic findings of Mendel and Morgan relate to the chromosomal basis of recombination. So offspring with a phenotype matching one of the parental phenotypes are called parental types. And offspring with non-parental phenotypes, which are new combinations of traits, are called recombinant types or recombinants. A 50% frequency of recombination is observed for any two genes on different chromosomes. Okay. So if we go back to Mendel's experiment and his model organism, which was the pea plants, we can also see that we had recombinant offspring that did not look like their parents. And also the other half was the parental type offsprings which did look like their parents. This was all due to crossing over that happened in prophase 1. Morgan observed that although some genes are linked, non-parental allele combinations are still produced. He proposed that some process must occasionally break the physical connection between genes on the same chromosome. That mechanism is known as crossing over of the homologous chromosomes. So knowing about crossing over during meiosis, we can take a look again at the parental fruit flies and see why we produced the recombinant offspring that did not look like their parents. Okay, and that was due to meiosis 1. So crossing over happens in meiosis 1. And more specifically, crossing over is going to happen in prophase 1 of meiosis 1. So what happens in prophase 1? Crossing over and crossing over. We can see the first crossing over here event. So VG plus and VG are going to switch between the homologous chromosomes, and we are going to produce two recombinant chromosomes. Sorry, that is not correct. So the switch happened here. My bad. Okay. So VG plus and VG were switched. So after meiosis 2, we separated those sister chromatids, and we get the two recombinant chromosomes at the end of meiosis in each of the eggs. So 4 of those eggs are going to have the parental and the non-parental chromosomes. And if we go through a test cross right here with the sperm, which have B and VG, we're going to get that ratio again. We have some recombinant offspring, but it's a lot less than the parental type offspring. Here's an animation of crossing over.
During prophase 1 of meiosis, homologous chromosomes pair up very closely, and corresponding parts of two non-sister chromatids may trade places. This process of crossing over creates variation by producing chromosomes that combine the genes inherited from two parents. Here the process produced a total of 4 genetically different gametes. There are many ways crossing over can occur. In humans, crossover events happen an average of 2 or 3 times per chromosome pair, greatly increasing the variation among eggs and sperm. Note that crossing over produces some parental gametes with chromosomes like those of the parents, and some recombinant gametes with a mixture of genes from both sets of chromosomes.
Okay, so this is a good video showing you the crossing over that happens during prophase 1 of meiosis 1, producing both recombinant and parental gametes. Recombinant chromosomes bring alleles together in new combinations in gametes. So that's going to increase diversity or variation. And random fertilization increases even further the number of variant combinations that can be produced. So random fertilization is referring to how sperm and egg are going to randomly pair up with one another. This abundance of genetic variation is the raw material upon which natural selection works. So the variations that end up being produced that are more favorable for survival are going to be selected against the non-favorable variants. Alfred Sturtevant, one of Morgan's students, constructed a genetic map, an ordered list of the genetic loci along a particular chromosome. And remember, loci is going to be where the gene is located on the chromosome. Sturtevant predicted that the farther apart two genes are, the higher the probability that a crossover will occur between them, and therefore the higher the recombination frequency. A linkage map is a genetic map of a chromosome based on recombination frequencies. Distances between genes can be expressed as map units. One map unit represents a 1% recombination frequency. Map units indicate relative distance in order, not precise locations of genes. So here we see a linkage map, and we see 3 genes. We have gene B, gene CN, and gene VG. Also, we can call them the 3 different loci or locus. And the recombination frequencies are here. So between B and CN, it's 9%. Between CN and VG is 9.5%. And between B and VG, we just add them together. Okay, so it's around 17%. So it's just an approximation. Genes that are far apart on the same chromosome can have a recombination frequency near 50%. Such genes are physically linked, but genetically unlinked, and behave as if found on different chromosomes. So basically, if you have a recombination frequency over 50%, it's likely that that gene is going to end up being transferred and swapped with another gene on a different chromosome. Sturtevant used recombination frequencies to make linkage maps of fruit fly genes. He and his colleagues found that the genes clustered into 4 groups of linked genes, linkage groups. The linkage maps, combined with the fact that there are 4 chromosomes in Drosophila, provided additional evidence that genes are located on chromosomes. Okay, so here we see a linkage map of the fruit fly chromosomes. So right now we're looking at chromosome number 2 out of 4. So the homologous chromosome. And I believe we're looking at whether the gene for the arista is going to be linked with any of the other genes on the same chromosome. And so the arista gene is the appendages on the head, so the antenna, and we can see the frequency, the recombination frequency is 0, and we compare it to the other genes on the same chromosome. So for the eye color, it's going to be 16.5%. So the arista and the eye color is probably going to be inherited together. And then next we have 48.5% for the color of the body, which is still below 50%. So the arista, the eye color, and the body color are probably going to be inherited together. So they are not going to be recombined. So they're not going to be exchanged. However, if we look at the latter parts, the genes that are further away from the arista gene, we can see how the eye color has a recombination frequency of 57.5, the wing shape 67, also the wing shape 75.5, or the curve of the wing, and the gene for whether you have brown eyes or red eyes instead of maroon and red, 104.5. So these genes, since they have a recombination frequency greater than 50%, these genes are probably going to not be inherited together with the genes on the left. So these genes will be recombined. So they're going to go through that crossing over event. So technically, all of these genes are on the same chromosome, so they will be considered linked genes. But the genes further to the right, they're probably not going to be linked. They're going to be unlinked if we look from the perspective of recombination. Okay. Large-scale chromosomal alterations in humans and other mammals often lead to spontaneous abortions, miscarriages, or cause a variety of developmental disorders. Plants tolerate such genetic changes better than animals do. So how do these, or what are some examples of these chromosomal alterations? We have non-disjunction. Non-disjunction occurs when chromosomes do not separate properly during meiosis. As a result, one gamete receives two of the same type of chromosome and another gamete receives no copy. So non-disjunction can happen either in meiosis 1 or meiosis 2, and that's going to lead to different results. So on the left right here, we have non-disjunction that happens in meiosis 1. Okay. So the tetrads had to separate evenly, but we can see right here that the tetrad did not separate, and this resulted in one of the daughter cells receiving more chromosomes and the other daughter cell receiving less. After meiosis 2, we're going to produce 4 of the gametes, and each of the gametes, as a result of non-disjunction in meiosis 1, are going to have 1 extra chromosome or 1 less chromosome. And just a side note here, we can see how non-disjunction it occurs during the separation of the chromosomes. So that would be anaphase 1 in this case. And during meiosis 1 and during meiosis 2, we would call that anaphase 2, where we separate the sister chromatids from the dyads. Okay. To the right, we have another scenario where non-disjunction is going to occur in meiosis 2 during anaphase 2. So if that occurs, we're going to get 1 gamete with an extra chromosome, another gamete with 1 less. However, if non-disjunction occurs in meiosis 2, we get 2 normal gametes. Aneuploidy results from the fertilization of gametes in which non-disjunction occurred. Offspring with this condition have an abnormal number of a particular chromosome. A monosomic zygote has only 1 copy of a particular chromosome, a trisomic zygote has 3 copies of a particular chromosome. So that would be similar to the pictures that we saw earlier. So the monosomic zygote is going to have only 1 copy, and the trisomic has 3 copies. Polyploidy is a condition in which an organism has more than 2 complete sets of chromosomes. Triploidy, 3n, is 3 sets of chromosomes. Tetraploidy, 4n, is 4 sets of chromosomes. Polyploidy is common in plants, but not animals. Polyploids are more normal in appearance than aneuploids, so that's very interesting. Breakage of a chromosome can lead to 4 types of changes in chromosome structure. So these are the different types of chromosome alterations. We're altering or we're changing the chromosome structure. Deletion is a removal of a section of the chromosome. Duplication is duplicating a section of the chromosome. Inversion is when we reverse the orientation, or we basically flip a section of the chromosome. And translocation is when we move one part of the chromosome, or we swap one part of one chromosome with the other part of another chromosome. So we swap sections of chromosomes between 2 different chromosomes. Okay. So here are some diagrams that show you the different chromosome alterations. We have deletion, where we basically deleted part D of the chromosome, and afterwards we don't have D. Duplication right here is when we duplicate a section of chromosome. So section BC is duplicated afterwards. So we have inversion is when we flip a section of the chromosome. So the order would be segments B, C, and D. But after inversion, D is going to come first, C second, and B last instead of B first. And finally, we have translocation. And this is the case where we have 2 different types of chromosomes. So let's say we had one type of chromosome right here and another type of chromosome right here, and we're going to basically swap section AB in chromosome, let's say 1, with section M and O on chromosome number 2. So we swap those. Afterwards, we're going to have section M and O in chromosome number 1 and section AB on chromosome number 2. Okay, so yeah, let me just label the hypotheticals. So this is chromosome number 1 and chromosome number 2, chromosome number 2, and 1. Okay. So deletion, duplication, and inversion all involve the same chromosome, but translocation is going to involve 2 different chromosomes, where we swap sections between those 2 different chromosomes. Alterations of chromosome number and structure are associated with some serious disorders. Some types of aneuploidy appear to upset the genetic balance less than others, resulting in individuals surviving to birth and beyond. These surviving individuals have a set of symptoms or syndrome characteristic of the type of aneuploidy. And so remember, students, aneuploidy is just a different number of chromosomes in an individual, and it's a result of non-disjunction. So non-disjunction in meiosis causes aneuploidy. Down syndrome is an example of aneuploidy, where an individual is going to have an extra copy of chromosome pair 21. It affects about 1 out of every 830 children born in the United States. The frequency of Down syndrome increases with the age of the mother, a correlation that has not been explained. So if we look at the karyotype here, we can see how each of the chromosome pairs, 1, 2, 3, 4, 5, and so on, they only have 2 chromosomes, each from the father and the mother. But if you focus on chromosome number 21, we have 1, 2, and 3. We have an extra copy of the chromosome, which results in the symptoms associated with Down syndrome individuals. Non-disjunction of sex chromosomes produces a variety of aneuploid conditions. Klinefelter syndrome is a result of an extra chromosome in a male producing XXY individuals. About 1 in 1,000 males is XYY. These males do not exhibit any syndrome. So non-disjunction of the sex chromosomes can result in different syndromes as well. Females with an extra X chromosome occurs with a frequency of about 1 in 1,000. They are healthy with no unusual physical features, though they are at risk for learning disabilities. Monosomy X, also called Turner syndrome, produces females with only one X chromosome who are sterile, meaning they cannot reproduce. It is the only known viable monosomy in humans. The syndrome Cri-du-Chat, I hope I pronounced that correctly, also called cry of the cat, results from a specific deletion in chromosome 5. A child born with this syndrome is severely intellectually disabled and has a cat-like cry. Individuals usually die in infancy or early childhood, sadly. Certain cancers, including chronic myelogenous leukemia, CML, are caused by translocations of chromosomes. So here is the translocation in CML. We have a normal chromosome 9 and a normal chromosome 22, but we can see how a section of chromosome 9 is going to be swapped with a big section of chromosome 22, and that results in our kind of mixed chromosomes. We call the translocated chromosome 22 the Philadelphia chromosome. There are two normally occurring exceptions to Mendelian genetics. One exception involves genes located in the nucleus, and the other involves genes located outside the nucleus. So we remember that there's going to be DNA in your mitochondria, called mitochondrial DNA. In both cases, the sex of the parent contributing an allele is a factor in the pattern of inheritance. So the sex of the parent matters. For a few mammalian traits, the phenotype depends on actually which parent passed along the alleles for those traits. Such variation in phenotype is called genomic imprinting. Genomic imprinting involves the silencing of certain genes depending on which parent passes them on. Here's an example of genomic imprinting in mice. So we have the LGF2 allele. The LGF2 allele is going to be expressed in males, but it will be silenced in females. If the LGF2 allele is normal, then we will have a normal-sized mouse. So the LGF2 allele or gene is associated with the size of the mouse. In the case of the LGF2 allele being mutated, so if the LGF2 allele on the maternal chromosome was mutated, then it would not affect the phenotype of the mouse. So here is an individual who inherited a mutated allele from the mother. But if the mother's allele was mutated, that wouldn't affect the phenotype of the mouse because the gene from the mother is not expressed anyways. However, if the LGF2 allele from the father was mutated, this would matter because the LGF2 allele from the father is expressed. So if a mouse inherited a mutated LGF2 allele from the father, then it would express a dwarf phenotype. so the mouse would be smaller. It seems that imprinting is the result of the methylation, the adding of CH3 methyl groups onto the cysteine nucleotides. So methylation is what causes the silencing. This is a field of genetics called epigenetics. Where outside influences are going to affect the expression of the gene. Genomic imprinting may affect only a small fraction of mammalian genes, and most imprinted genes are critical for embryonic development. Finally, extranuclear genes, or cytoplasmic genes, are found in organelles in the cytoplasm. For example, the mitochondria, the chloroplasts, and other plant plastids carry small circular DNA molecules. Extranuclear genes are inherited maternally because the zygote's cytoplasm comes from the egg and not the sperm. The first evidence of extranuclear genes came from studies on the inheritance of yellow or white patches on leaves of an otherwise green plant. Some defects in mitochondrial genes prevent cells from making enough ATP and results in diseases that affect the muscular and nervous systems. For example, mitochondrial myopathy and Leber's hereditary optic neuropathy. So genes that are located not only in the nucleus but in the mitochondria are also important. It may be possible to avoid passing along mitochondrial disorders. The chromosomes from the egg of an affected mother could be transferred to an egg of a healthy donor, generating a two-mother egg. This egg could then be fertilized by sperm from the prospective father and transplanted to the womb of the prospective mother.